Every time we decide to have children, we are unconsciously doing a kind of natural genetic engineering. We know our children will be like us in some ways, but we can't predict exactly how they'll turn out. Imagine a world in which you could predict, or even choose, particular characteristics for your children. Why is such an idea so unsettling?
Some mothers are better than other mothers, and some fathers are better than other fathers. And so if you try and have the best DNA possible with the most intelligent people, the best at sport, the friendliest people, and you have the best mother and the best father's genes, and you put them together, yes, you'll get the perfect person. Hopefully.
Do you think it'd be a good idea if we used genetic engineering to make people different from how they would naturally be?
I don't think you should have a prejudice against it, but I think you should be careful when you do it. You shouldn't experiment just with people, because it would be a waste of their lives.
What do you think, David?
Well, I don't think it would be good to use genetic engineering, because then everyone would be kind of really brilliant at something or other, and there would be no competition, like say a sport. And the father was say cricket. Father was a cricketer, so he chooses his son to be a brilliant, absolutely fantastic cricketer. But they'll slowly become more and more better things. It wouldn't be just natural ability to play these things. Just be chosen to be good at something. It wouldn't actually be good at it.
Well, I don't think that's right. Why wouldn't you be good at something if you're So, cricket, your cricket example. If your father's very good at cricket, he buys, say, from this shop which sells them DNA, the best, say Ian Botham's cricket DNA. Okay, the part of the DNA which makes Ian Botham good at cricket, and then he puts it into you, and so you're fabulous at cricket. Why does it mean you're not fabulous at cricket?
Well, it could be, say Ian Botham isn't your father. Your father's just interested in cricket. He likes the idea
You wouldn't have a father, that's the point.
When I was at school, I read Brave New World and was alarmed by the idea of rows of programmed people being produced by a central hatchery. But I found it hard to be sure how far my resistance was just emotional prejudice and how far my objections were real. As biology catches up with fiction, it's becoming more important that we all sort out which of our feelings are irrational and which justifiable. For the first time, the act of conception can be observed and achieved in the laboratory. There are concerns that this may lead to manipulations on the human embryo. Already, frozen sperm is big business. Artificial insemination by mail order is now with us. Human eggs and embryos can be frozen for years, when they will still be fresh for implantation into a mother. One woman's egg may soon become another woman's baby. The techniques have all come from animals, and have all gone further in animals. Genetically identical clones of frogs were produced in the mid-50s, but the cloning of mammals was considered too difficult. Then, last year, Cell magazine announced that 3 mice had been cloned in Switzerland by manipulating their embryos. Mouse eggs are as small as human eggs, so in theory the same could be done with humans. The same team have even produced virgin birth in mice. The first steps are now being taken to make the womb redundant. At Cambridge, young rats are being removed from the placenta and grown up outside the bodies of their mothers. So far, they can be made to develop in a laboratory up to a stage where their organs start to emerge. Manipulations are no longer confined to whole cells or their nuclei. Now, individual genes are being moved around between species, from bacterium to rabbit, from rabbit to mouse. In fact, there's nothing to stop a gene from an orange incorporating successfully into the genetic material of a duck. Computers can now build up genes from 4 basic chemicals taken off the laboratory shelf. Every month, longer and longer sequences of synthetic genes are being stitched together by a machine. Scientists are confident that, within 20 years, they will be able to work out the chemical structure of all the genes in a human being, a complete map of all the chromosomes. Human genes have already been injected into the cells of other species. At Yale, a fertilized mouse embryo is the target. The human gene will become an integral part of the young mouse and will be carried through to subsequent generations. These experiments have all been on animals, but scientists realized the possibility, not always faced, that the same techniques might one day be applied to humans. Clement Marcotte is professor of biology at Yale.
There are many people in our society who feel that in the revolution in biology going on with genetics, that we are maybe giving human beings a capacity to do things which we're not really prepared to handle adequately. We can shortly preside over our own evolution and make new kinds of individuals, including new kinds of people. And are we socially and intellectually prepared to do that? That's a very serious question as to whether that's true or not.
Now, I've no doubt that there are a lot of practical difficulties on the way before we get to the point where we can choose what genetic characteristics to give to people. But nonetheless, I think there's a case for carrying out a thought experiment of asking what we would like and not like, what we should and shouldn't do, if ever we did get to this advanced stage of being able to choose people's genes. The case for this thought experiment is partly that I think too often in our social response to new technology, we've gone through a rather terrible history. We've gone very rapidly from an early stage when scientists have said, oh no, this is not going to be practical. Of course this won't happen. There are overwhelming technical difficulties that you laymen will never understand in ever bringing this about. We've gone very rapidly from that first stage to a sudden disturbing transition to the point where people say, well, actually we've had the breakthrough now and we're doing it, and it's really too late for any discussion and it's too late to stop it. These predictions about impossibility are just simply very often wrong. I mean, the famous case of the astronomer royal in the 1950s, who said that the very idea of space travel was utter bilge. Or Rutherford in the late 1930s, who took the view that the kind of work he was doing in atomic physics couldn't possibly have any practical application. When he was asked about this, he said it was simply moonshine. And about 10 years later, the atomic bomb was dropped. What I'd like to do is take what I assume is true of most of you, that you share the feeling I have of fairly deep feelings of disturbance, even perhaps amounting to revulsion, at the thought of human beings starting to engineer their descendants.
Nice and comfortable on the cushions. Gently relaxed. Wriggle around until you are comfortable. And close your eyes now, but remember, in labour, keep them open. Let the muscles go. Give yourself lots of room. Give the baby lots of room. Sigh away the outward breath through your open mouth.
I think you should be happy with what you get, and you shouldn't try to be some sort of creator of some I suppose like the Nazis tried to do, some sort of master race or some race or some kind of human being you would like to have. I think you should accept nature.
I think I'd do anything to make sure the baby was healthy, but I'd find it rather arrogant to try and impose some set of characteristics. I think I see being a parent as sort of bringing out the individual's qualities, and if you're going to begin by saying what you want an individual to be rather than letting the individual be, that's getting things off on a rather bad basis.
I think we're arrogant in just deciding how we're going to bring up children. The assumption that the things I want my child to be are right is an arrogant assumption in itself.
Don't you think we're interfering with nature all the time? Through medicine, through schooling, through machinery?
Yes, but then you're not doing something once and for all. You've got other influences, and other people that can have counter influences and perhaps mitigate the effect that you have on the child.
I don't see any reason for saying that it's perfectly all right to mould people environmentally, but not all right to do the same by genetic means. And a point perhaps worth making in this context is that social policies we've already embarked on are in fact changing the genetic composition of future generations now. Every time we do anything like set up the National Health Service, we make a difference to who can afford medicine, as a result who can survive, and as a result who survives to have children. And as a result, a different set of genes enters the next generation from what would have happened if we hadn't set up the National Health Service. But nobody, as far as I know, says, oh dear, we hadn't thought of that, we better not have a National Health Service. The fact is that it's hard to think of any social policy with broad impact that doesn't have some sort of, even if unintended, eugenic impact. If we consider the genetic screening programs that have been introduced in the last few years to detect various kinds of abnormality in the fetus, we find that, on the one hand, these screening programs offer tremendous hope that we don't have to have children born who are abnormal, but on the other hand, they arouse a certain opposition. With genetic screening, we are already starting to select who is born. Take the case where a single abnormal gene produces a severe blood disorder.
About 3 years ago, when my husband and I decided to start a family, I went along to my GP for a German measles vaccination, and at the same time my GP thought it wise that I have a blood test, and my husband also had the blood test, and this showed that we were both carriers of thalassemia. This meant that if we had a child, we had 1 chance in 4 having a thalassemia child. He explained that there was an antenatal diagnosis test available which detected the thalassemia child at about 20 weeks of pregnancy. And unfortunately, the first time it was a thalassemic child, so we had the pregnancy terminated.
When you were taking that decision in advance, what sorts of things weighed with you to make you decide that way?
Well, the doctors explained the treatment available at the moment for thalassemic patients, and personally I wasn't really happy to bring a child into the world with the treatment that was available. There's no known cure that I know of. And, I couldn't stand the thought of my child having blood transfusions every 4 weeks for all of his life, and injections and deaths for all pumps. You know, it's very, very uncomfortable for the child.
And then you got pregnant again?
Then I got pregnant again, yes, and unfortunately the second time round was exactly the same. But I found it was much easier to make the decision the second time round for the termination. Having gone through it once, I simply couldn't have not gone through it a second time.
Were you relieved to hear that there was a test of this kind?
I was, yes, because had there not been a test, I'm sure I wouldn't have gone in for a child, so really we wouldn't have any children.
So Mary here wouldn't
Mary wouldn't
exist.
be here, no.
If the screening program is okay, then surely genetic engineering to knock out the disease must be acceptable, because if it's all right to kill the baby because it has this disorder, surely it's preferable, even if we're in favour of abortion, surely it's preferable to cure the disorder in the fetus rather than have to kill it and start again. So we already, if we accept the screening program, are going to find it hard to resist negative genetic engineering. Negative engineering being simply genetic engineering used to knock out recognisable categories of medical disorder. But where does the slippery slope go after that? Are we likely to move on to what's sometimes called positive genetic engineering? Positive engineering being the use of these techniques to bring about improvements in people who are otherwise normal, people who are not in any way medically defective. Say to make people stronger, or make people kinder, or make people have a higher IQ. Some people say, well, it's all right to accept the negative uses, but we really mustn't cross the boundary towards the positive, because there we're starting to do things which we really shouldn't do. My unease, though, about trying to draw the boundary there, if somebody says, let's have a principle, negative genetic engineering is okay, positive genetic engineering has to be ruled out. My unease is in part that I can't quite see exactly what the reasons are for drawing this distinction, and in part my anxiety comes from the thought that the boundary may not be that much of a sharp one. Take something like depression, which may very well turn out to have some genetic component.
I think certainly in health terms, if there are genetic, and particularly in psychiatric health, if there are genetic things that are found, then I think if you've known someone who's suffered psychiatric illness, to be able to spare someone else that in future, it's worth considering.
So you're inclined to draw a boundary between knocking out disorders, whether physical or psychiatric, and choosing positive characteristics like kindness.
That's probably where I would, yes.
Are you sure there's a sharp boundary there to be drawn?
That's why everything has to be a decision that's calculated at the time on the risks involved. And
You'd knock out a genetic disposition to clinical depression.
I think if it were possible, it's only a form of preventive medicine after all, and we take preventive medicine against other illnesses.
But if that's okay, is it so clear that there's a sharp boundary between doing that and bringing someone up from, say, fairly low down in the normal range to fairly high up in the normal range? If we make normal people that much more cheerful, are we really crossing a very sharp boundary when we do so?
There's a certain amount of depression that is just not feeling particularly happy at the moment, that is very different to a clinical depression. And yes, I realise the dividing line is fine, but then psychiatrists make a decision about the dividing line when they decide whether to treat someone as an illness or just as someone who's not very happy today. And I think you couldn't take out everything that wasn't 100% perfect about being alive. Or perhaps you could, but then I'm not sure what kind of world we'd have reached.
Do you think it would be a good idea to alter normal people's genes to make them generally more cheerful? Why not? Oh, you disagree with David. Why do you think no?
Well, because part of life is having the ups and downs, being sad and happy, If you're just kind of happy, no depressed, never get depressed about anything. Wouldn't really be a person, it wouldn't be a human being, it would be utterly altered. You'd just be happy all the time.
Surely some people would never get depressed.
You think it would be good to engineer people so that they're never depressed? Why do you think that?
Because it can do no harm. It's all very well saying it's a different person, so it doesn't matter if it's a different person. It means that person's changed for the better.
But supposing I tried to use the same argument for making people more intelligent by saying they might not be exactly the same person, but it would be much better.
Well, that doesn't follow, because being more intelligent doesn't mean you have you may have more chances in life. Like disabled person doesn't have as much chances as a person not disabled, but intelligence, okay, it's nicer. I think quite a few people agree. It's nicer to be normal, when not handicapped or anything like that. But with intelligence, it can be perfectly nice to be a bit stupid. Does it matter? Does it matter if you're not the same person?
Yes, it does. Because it's not the same brain. It's not the same you.
Doesn't matter.
Yeah,
Someone else.
the person before, or the person that was going to be formed, if he knew it, then, or she knew it, they might be, upset.
I think people feel a deep revulsion, which often comes out in the phrase, who are we to play God in this matter? Who are we to decide the future of the human race in this sort of way? This is partly, I think, the objection that when one starts to focus on who would take the decision, some pretty disturbing implications can arise. Suppose we imagine the decision being taken, say, by a group of scientists. I think what we're worried about is the feeling that that group of scientists, say, a dozen very distinguished fellows of the Royal Society, what seems worrying is that however admirable they are, we don't want the limits of future human variety to be set by the limits of the imagination of this dozen or so distinguished scientists. And I think the position isn't actually that much better if one says, well, let's broaden it out. Let's have the future of mankind decided by some committee consisting of 2 MPs, a civil servant, a doctor, a lawyer, and a clergyman. I don't feel that much reassured by the thought that that's what's going to happen. But I think it should be said that we don't have to imagine the decision being taken in a totally centralized way. It doesn't have to be some committee, some central group. There is what's sometimes called the genetic supermarket as another possibility, the idea that parents might choose the characteristics that they would like their children to have.
Here are your 6 fertilized eggs in the cryobank, 2 male and 4 female. And as you requested, Pat, they all come from your 3rd superovulation from the batch that produced Lily. And the sperm we injected them with all came from your May donation, Conrad. Now, the fertilizations were all very successful. And the zygotes have all been gene-typed, and they're looking very good, with the one exception of female zygote 5, which has got an abnormality on chromosome 6, and that could cause major developmental problems, so we won't be using that one. Now, are you quite sure? I mean, you've already got 2 girls. Are you absolutely certain you don't want a boy?
Oh, yes, quite sure. We really do want another girl.
Yes, definitely. Right. You've had a chance to view the data at home.
Yes, we've narrowed it down to zygote 3 and 6. We're not really sure which one to choose.
What sort of characteristics were you thinking of?
Well, we definitely don't want to tamper with the physical side of things in any way.
No, except that we would like her to have my father's red hair.
Oh, well, that's easy. We can make her homozygous on the 3 hair color genes. What about her character and emotions?
Well, yes, there are a few things we'd like to have modified if possible. We'd like to reduce shyness and susceptibility to depression without necessarily damaging any artistic potential. Also, we'd like her to be musical, and if possible, also we want her to be ambitious.
Of course, we want her to be as healthy as possible.
Well, let's have a look at 3 and 6 then. Yes, I think we should concentrate on 3. I think 6 is going to require too much work. Well, let's see if we can make a budding musician out of her. I've had very good reports from the Chinese about a new gene cluster. Let's see if that's available. Yes, it is. Good. Now, deleting the shyness and depression without affecting the artistic ability is a bit more delicate. But you had quite a good experience with the Bash Kremko deletion with Daisy, didn't you?
Oh, yeah, she seems reasonably extrovert. Certainly her painting's very promising.
What sort of things do you like painting, Daisy?
My family.
Do you? Well, next time you come to see me, why don't you bring me a picture of your family that you've painted? Then I can put it up on my office wall.
Yeah.
That'd be lovely. Well, the only problem seems to be with ambition. The GMAP committee are currently re-evaluating the societal impact of the Bibbs-Murdoch configuration, and it has been temporarily withdrawn. But I will just check it for you. Yes. Well, I'm sorry, but there should be no problem with the other changes.
Well, I think we can live with that.
Right. Well, then I'll work on the fine detail, and that'll give you a week or two to think about things. And then when you're quite satisfied with the final composite, we'll go ahead and start the manipulations. Do you want to have the child yourself?
Oh, yes, definitely.
Right. Well, then I'll go ahead and book your implantation appointment as soon as we can fit you in.
Thank you very much.
David, suppose that we could have chosen to make your genes different in some way. Is there any way you think it would have been good if we had?
No, I don't think so. You know, okay, you could have made me better at music or something like that, but I don't think that would be good, because then how do you know it would be me? It would be a different person, because I'm not very good at music. But if you programmed me to be good at music, then it wouldn't really be me. It wouldn't be the same David.
Perhaps before going too far with these futuristic speculations, it's worth coming back to present reality. I went to the Department of Zoology at Oxford to ask what technical obstacles stand in the way of human genetic engineering.
So this is what a well-organized laboratory looks like for the analysis of the genes once they're incorporated into the mouse.
Dr. Chris Graham probably knows as much as anyone about the current state of genetic research.
Chromosomes and finding out what's happened to the genes. And then if we go over here, we have the famous micromanipulator, which was used by Gregory Peck and the boys from Brazil. And it's the device which we use for injecting genes into the mouse egg. And what's involved is to take a large syringe like this and to blow through it, and then the genes come out of this very, very fine pipette at the end and lodge in the nucleus of the egg. I think there's a very general view at the moment that what genetic engineering can do for you is to introduce genes and then completely ginger up the human character which emerges in the baby. This is a terrible misconception. On the one hand, it's true that we can squirt genes into animals and that those genes will integrate into those animals, but we can't direct where they go. We don't know what controls their expression, and we're completely uncertain about the efficiency of that process. And so on the one hand, there are technical problems simply involved in introducing genes into organisms. The other problem is that the human character is a much richer thing than simply a product or readout of its genes. It develops through interactions with the uterus, with the environment, and is realized through educational systems. So there's no way in which you can take a genotype associated with low IQ and hype it up by squirting in a gene to be a highly productive, very intelligent, clever person.
Could I ask you about the first of those difficulties? I'm not committed to the view that there aren't any practical obstacles which may stop us, but I'd like to get clear just what exactly you think they are.
Perhaps I should say we know so little about human inheritance. it's a reasonable guess that there are over 1,000 genes in humans. A catalogue today of the number of human genes we know something about, inherited traits, amounts to 3,000. The structure of the genes involved in controlling these traits is probably known for about 4 or 5. So that our ignorance of the inherited characters of human beings is immense. And what this makes it extremely difficult to do is to predict how an introduced gene would behave with the pre-existing genes, and how the pre-existing genes would behave as human culture and environment changes with history.
Do you think that our ignorance will always be with us, or might we one day have the kind of knowledge which at present we don't have of all the different human genes?
I don't think our ignorance will always be with us, nor do I think it'll be as great as it is now. But it's important to remember that if one sat down and wondered, for instance, how to create a child who was good at running the 5,000 meters, what would one do? Well, what one would do would be to find a happy couple who wish to have a child who can run the 5,000 meters. And then we would hope that genotype was heavily involved in this characteristic. And then we'd ask ourselves, well, what kind of genes do we need to make these people run so fast? And what we might do is to inquire into the genotype of Steve Ovett and Sebastian Coe. Well, we know very little about those genotypes because they haven't bred and we haven't analyzed their future ancestors. So what we might decide to do would be to sequence their genes. We'd make a DNA library of Sebastian Coe's genes and Steve Ovett's genes. Now, a minimum estimate would be 6,000 scientists working for 40 years to find out what the genes were like in each of those individuals. And the happy parents eventually have a child, and there is a hope that this will now run fast. But wait a moment, maybe they've decided to hold the Olympics, this time in Mexico City, at a different altitude to the altitude at which Steve Ovett and Sebastian Coe ran so well. And we've got the wrong genotype for Mexico City, although we might have had the right genotype for Moscow or some other place.
I wonder whether you can be confident that future scientific development won't allow us to take some shortcuts. Your argument, it seems to me, is that the discussion is pretty unrealistic because you think that the possibility is extremely remote. But that's something which it seems to me in principle really rather hard to argue for, because all you can do is appeal to our present state of knowledge and extrapolate from that.
What I think is important to say is that if it really was a wish for parents to have children with predictable genotypes, then one certainly wouldn't get involved in squirting genes into their eggs. What one would do would be to find an individual which had the characteristics which the parents wanted and attempt to clone from that individual. Now, I must say right away that we can't do this at present. We can do it with mice using embryonic nuclei. But the important point is that if you want a nipper with a predictable genotype, you would do it by cloning, and you wouldn't mess around with gene supermarkets or any other device which involved genetic engineering.
The idea of human clones is repugnant to most of us. But why? After all, identical twins and triplets are in fact nature's clones. These brothers have just celebrated their 21st birthdays together. They went to the same school, were good at the same subjects, and enjoyed the same hobbies. Andrew, Martin and Paul Tunney still share the same home and all work in the same factory. Genetically, they're identical. But are they really that similar? When you were children, it didn't make you feel that you were less of an individual person?
Not really, no. We did stick together, but we did have our own sort of individuality.
Yes.
You know, my mum did notice differences, you know, in likes and dislikes.
Now, how would you have felt if instead of being 3 of you, there'd been 30?
I'll have to ask my mother. Yeah, how should react to that? I think 3's enough.
A family with triplets is very attractive. But the idea of making large batches of identical people is disturbing, because the interesting differences between individuals might disappear. We may even have a slightly more fanciful idea when we think about cloning. We may wonder what it would do to relationships if a member of a clone had special relationships with other people. I mean, what would it be like, for example, to fall in love with a member of a clone? Isn't there a danger that we might not fall in love with the individual person, but, as it were, fall in love with the whole batch?
They always sort of asked at work things like, which one are you going out with tonight, you know, so we can swap.
At first I wanted to go out with Martin, but I couldn't tell the difference really anyway. Sometimes I'd walk up to Martin and say hello Paul, and it'd be Martin. And I'd walk up to Andrew and say hello Paul, and get them mixed up, but no, I don't.
I think I was fascinated that there was three of them at the beginning, but now I don't know really, just grown to like Martin.
If there were 30, do you think you might start to feel that it would be nice to go out with any of them and you didn't very much mind which?
I might be tempted. I don't know.
Would you be tempted?
They would, yeah, being to so many of them.
How about you?
Yeah, I probably would.
We all read many science fiction books, if you're interested in science fiction, which are based on the idea that a man has been cloned. Apparently it's always a man rather than the woman that gets cloned. Well, at the present time, we haven't any reason to believe that adult cells, taken from an adult man, could be used successfully to clone him, except possibly for the spermatogonia, which in fact do have all the genetic makeup and we know are not irreversibly differentiated. Well, if a man is to be cloned, and if spermatogonia are to be used, it may well be that he will have to sacrifice one of his testes in order to be cloned, and that might be a proper moral price to pay for that cloning.
No man has been cloned so far, but there are some very sophisticated operations now going on at Cambridge. At this animal research station, they have made sheep clones by splitting embryos, and now they can produce sheep with 4 parents.
Okay. And then...
That's where you collect them?
Yes. The embryos will be in the oviduct. We actually flush a little bit of the uterus as well, because it's difficult to get a needle into the oviduct. So now I now have the needle in the very tip of the uterine horn.
What are you going to do with these embryos you're taking out?
These embryos we are going to use to study early embryonic development. We've got 2 donor sheep, and we're going to take the cells from one embryo, from one of the donor sheep, and mix with cells from an embryo from the other donor sheep, and then examine how the composite embryo, which is formed in that way, develops.
First, they tease out the young cells in each embryo. Then they suck up one cell from each and introduce them together into a new foster home. The man-made embryo is then put back into one of the sheep, where it will develop normally. The resulting creature has 4 parents and is called a chimera. So one of these is a chimera, is it?
Yes, that's right. See if we can get hold of him. So this is the animal And you can see that his color markings are unusual in that he's generally a black animal, but he has these grey patches in the face, and he's also got...
I think quite a lot of people, when they think about that kind of experiment, start to feel a bit disturbed, start to feel that we're tampering with nature in ways that seem to lead to rather alarming things. But even if we aren't very worried about mixing genes in that way within a species, the next step of mixing genes from different species, I think, seems to many people really pretty disturbing.
These two animals, of course, are a goat and a sheep. Normally, goats and sheep are not able to interbreed, but interestingly, if you mix cells from a goat embryo with cells from a sheep embryo, then they're able to combine and form a viable embryo. And although this is not a crossbred, but a sheep-goat chimera, such an embryo can go to term.
Can we see one?
Yes, so this animal here is a sheep-goat chimera. We've made several of them, but this one is the most striking one. And you can see in this animal, which I must emphasize is not a crossbred, but a mixture of sheep and goat cells contained in just one animal. And you can see how he looks like a goat, but he's got wool in several areas on both sides. His horns, although he's knocked them off, are something in between sheep and goat horns. And there are several other characteristics of this animal which show that he is in fact a chimera.
What for you is the point of producing a chimera like this?
Well, the point, as far as we're concerned, is mainly to examine how the mechanisms of early development are similar in these two species, or if they're dissimilar, then to show that this is the case.
Could I ask what to many people is the rather disturbing science fiction kind of horror? What about humans and other species with whom they have a common ancestry? Is, from a technical point of view, this kind of thing possible?
Well, first of all, one has to realize that it's not just a matter of constructing an embryo, but it's also a matter of finding a suitable recipient. And secondly, I think that it's very unlikely that one could find people who engage in that sort of research. Now, whether it would be biologically possible, of course, one wouldn't be able to show unless experiments were carried out.
Is there any reason to think that it's unlikely to be possible?
Well, I think that it might be possible or might not be possible. I don't think that is the point. I think that the point is that whereas in animals, such as the sheep and the goats, there are some very good reasons why one should undertake that sort of research, it would be very difficult to see the purpose in scientific terms, or indeed in other terms, of a similar experiment on human embryos.
We do now put tissues from different animals into human beings. We put valves and hearts that are taken from pigs, and if we could succeed in curing the problems of rejection, we might even put pig hearts in to replace human hearts. And probably none of us would object to that, even though such an individual would be a sort of chimera. But if we were to make the chimeras from scratch, by putting embryos together, we would certainly produce a very odd creature, and one that would confront most human beings with very difficult ethical problems to resolve as to how they are to regard this creature, whether as human or as animal.
When we think of genetic engineering, it's the extreme cases, like man-monkey mixing, that worry us most. There's a fear that someone might actually try to make such a creature. We feel affronted. One of our deepest taboos is being challenged. But the defense of this boundary between species is not just a matter of taboo. Since we treat animals in ways we wouldn't think of treating any human being, there's a danger that we might extend our present exploitation of animals to subnormal members of our own species. This risk makes it very hard to see how such an experiment could ever be justified. But even apart from extreme cases, the idea of making any genetic improvement in people arouses our resistance. Partly for the good reason that there are very real dangers. We may make mistakes that would be hard to reverse. We would need some pretty clear benefits to justify the risks, and obviously any attempt should be made only with extreme caution. But I think perhaps there are some benefits worth setting against those risks. There's a thing Anthony Storr says in his book on human aggression. He says, with the exception of certain rodents, no other vertebrate habitually destroys members of its own species. The sombre fact is that we are the cruelest and most ruthless species that has ever walked the earth. Now, we don't like to think that about ourselves, and I'm sure it's not the whole truth about us. But when we think about the history of the 20th century, when we think about the wars and other horrors that are the products, if you like, not just of our aggression, but of our tribalism, our tendency to obey, our conformity, our willingness to believe rather than doubt, it's not obvious that it's going to be at all easy to eradicate war. There is, of course, the cheerful view that all we need to do is establish socialism or reform the United Nations or whatever else it may be, and war will fade away. But can we really be so confident that by these environmental means we're going to be able to do this? I'm not for a moment suggesting we should just despair and, as it were, say we can't do anything about war till genetic engineering comes. That would obviously be a negative and appalling attitude to take. But I think we have to face the sombre possibility that however much we try by environmental change and social change, it may be that the disasters that have characterized our history won't be changed by those methods. And if we do find that, and genetic engineering is available, I think we may, in this respect particularly, be wise to redesign ourselves if we can.
I think we should just choose for other people's lives. I think it should just be kind of, you just...
Potluck?
Yeah.
Why do you...
Better to choose than to have potluck.
Why do you think that?
Because, David, otherwise nobody chooses. You're preferring that they could be deformed, they could be spastic, they could be very, very stupid.
Yeah. But
rather than they'd be nice, they'd be friendly, and they'd be intelligent.
Yes, I prefer the thing where they could be stupid or anything. At least it would be my child. It wouldn't be, it would be just as much anyone else's.
All you're thinking about is theory about, but you're not thinking of the consequence. You're just thinking of what you would prefer it to be, not the actual consequence.
Daniel, aren't you a bit worried about parents possibly making the wrong decisions?
Yeah, I think that. Yeah, I was assuming that everyone, it was definite that it was safe and it was okay.
But even if it was definitely safe, isn't there a danger that too many parents would choose the same characteristics and we'd live in a more boring world?
That's a disadvantage of it, and that's a reason why perhaps we shouldn't do genetic engineering.
So which do you think on balance, that we should do it or that we shouldn't?
I think we should experiment first, and then we should see. I think, well, I'm not sure.